My name is Nabiha Saklayen. I'm the CEO and co-founder at Cellino. I'm a Harvard-trained physicist with a passion for bringing cutting-edge physics-based technologies to regenerative medicine. And I'm just honored and thrilled to be sharing our work with you today. Our mission at Cellino is to automate the process of generating autologous iPSC-derived therapies. We do this in a very unique way where we merge together machine learning with laser physics and stem cell biology. I'm excited to tell you more about that story. We're developing a unique platform to reach the promised land of regenerative medicine, which really is about how do we make patient-specific autologous cell and tissue therapies at scale, because today it's a very expensive, labor-intensive process to make such therapies for patients because the iPSC generation manufacturing process is very, very hands-on. So I'll show you a quick video of one of my iPSC scientists doing iPSC reprogramming and generation and cleaning a more manual way. So you'll see this. And the way this entire process starts, you have these cells in a well plate or a petri dish, and essentially you start by looking at these cells under a microscope, doing visual inspection and deciding which cells are high quality and low quality. Then the scientist, in this case, will go in and use a pipette tip to scrape away cells that look bad by eye. Of course, in this situation, Mariangela is really good at this, so she's still able to remove all the cells we don't want around the iPSCs, but it's not a super precise process. You definitely do not have single cell resolution in this case. So combining the visual characterization with the manual removal of the cells, it's not a very scalable or reproducible process for the most part. And this is how the industry does this process today, manually. And in order to make iPSC-derived therapy scalable for patients in a cost-effective way, this entire process really does need to be automated. And Cellino is doing that in a very unique way. How do we do it at Cellino? We start by imaging lots of cells in bright field and in fluorescence. And then we take these images and feed them into machine learning algorithms to recognize the high-quality cells versus the low-quality cells. So you're removing that human step. We've created an entire software infrastructure to run the system. And then we have a hardware system, which is our laser processor. And the laser will target individual cells on the plate that we want to eliminate, that the machine learning algorithms have deemed as low quality, quote unquote. As you can see, the laser system has single cell resolution. And that's really exciting because it really allows you to make the highest quality tissues you possibly could. Our platform consists of the AI-driven laser processing system, which is a hardware system. And we also have these custom well plates that play an important role in the entire process. We've designed the platform to really be an end-to-end solution for autologous cell therapies. We start with donor cells, and then we reprogram them and do clonal selection to make high-quality iPSCs. Then these iPSCs are differentiated to make both progenitor cells or highly mature cells for the cell therapy application. Of course, depending on the application, you might want to transplant different stages of cells in the patient for your best engraftment results. And what's really interesting is because we're integrating AI-driven approaches at every stage of the process, it's also going to be possible to look at an iPSC and know, is this iPSC going to become a high-quality progenitor or mature cell in the future or not? And if you like it, you can keep it. If you don't like it, you can remove it with the laser. So the power of using machine learning and automation really opens up endless possibilities in how we improve the process and the outcomes of the process as well. So now let's take a look at how this actually works. And the first step, of course, is training the machine learning model. And the way we do it is we take pairs of bright field images with pairs of fluorescently tagged images. In this case, it's a nuclear stain for the iPSCs in DAPI. And we feed these larger data sets, these pairs of larger data sets into the neural net to train it. Once the neural net has been trained, You don't need to feed it any fluorescent images anymore. You just need to feed it a bright field, label-free image, and it will essentially recreate what the corresponding fluorescent image should look like, as you can see in this demonstration. It's really nice because you can train the models using fluorescent assays that are terminal to the cell, and essentially you wouldn't be able to use the cell for a cell therapy product after doing that assay. But in this case, you can train the model with those types of assays, and then the final processing is always done in a label-free environment. And then, of course, the final steps for the machine learning algorithm, of course, decide which cells we care about, detect them, decide which ones we care about, and then output coordinates to process with the laser. So output the coordinates that we want to target on the plate, which cells do we want to keep, and which ones do we want to remove. Those are the ones we're interested in. Now, moving on to the laser, I've been talking a lot about that, and I'm a laser physicist by training, and this is how I got involved in this entire incredibly powerful, inspiring industry. But the laser does two things. One is either it can kill and remove cells you don't want, or it can create temporary pores in the cells for the delivery of payloads, such as CRISPR-Cas9, if you're interested in that. And it's very elegant the way it works. Essentially, we shine laser light onto the bottom of these custom well plates that we make at Cellino and generate cavitation bubbles at the bottom of the plate. And these cavitation bubbles can either kill the cell, remove the cell from the plate, or they can deliver cargoes into the cell. What's really nice is this is one of the only methods out there that can do single cell targeting, whether it's for removal or delivery, opening up this really wide range of high precision activities you could do with IPSCs for the first time, which we're very, very excited about. Okay, so now that you've seen how the machine learning training works, how the laser works, let's look at an example of how this process would go. So once the model is trained, then essentially the system will take a label-free image like this one over here, detect which cells are where, and create this density map of where the cells are, and then also create a desired density map. And in this case, you can see it's this web-like structure of cells. This is what we're targeting essentially. And then it'll create a keep and kill map, essentially. It'll decide which cells to keep and which ones to kill. Once the algorithms run through all of that, we can take a look before and after. And before, you can see denser cells here in this white square. And then afterwards, you see these beautiful web-like patterns, which means we have eliminated the cells that we wanted to get to this desired density. And when you zoom in closer, it just confirms that we have single cell resolution, as you can see. And we also have the ability to intentionally offset the laser should we choose to protect a certain population of cells. That's also very, very possible. Now, we've talked about how do we recognize where the cells are, use the laser to manage the density on the plate. The next thing to think about is what do we do when we want to train other additional features? And there are a lot of interesting features in IPSCs. So we take additional data sets. And here, the data sets are being taken to classify which IPSCs are pluripotent and which ones are not. And here we're taking bright field images with pairs of fluorescently labeled cells. And they have two stains on them, a nuclear stain, which is the blue one, and the green is OCT4, which is a pluripotency marker. We're teaching the model to classify which cells are pluripotent, which are the good IPSCs, and all the other cells are the spontaneously differentiating ones. And what we'll eventually be doing with this data is to remove those spontaneously differentiating cells so you're left with really high quality IPSCs on your plate. Now that we've looked at some of the examples of using image-based data to train machine learning models, let's take a look at what's next. And essentially what we're interested in doing at Cellino is forecasting which IPSCs are being successfully reprogrammed during this entire process. In order to do that, the data sets look a little bit different. So essentially you have to take time series data. So you take data sets at different time points during the process, and then you pair on top of it different data sets like qPCR, FACS, and trilineage differentiation. It just gives you richer biological data to go off of. So when you feed these training data sets into a neural net, what we should be left with is a model that can predict, okay, this colony over here is going to become pluripotent at the end of the process, and we only focus on keeping that one and we eliminate all the other ones. It's really powerful to see what is possible when you start bringing together the best tools that we have at the table with machine learning, with laser processing, and of course on the stem cell biology side, we've made tremendous progress over the past decade. The other thing that we've built into our platform is the ability to take really high quality data. We've really embraced the power of big data. And every time we take images of cells, every single cell is turned into a data point in our Cellino database, giving us access to massive amounts of data that we can use to make our algorithms stronger, and also is actually super powerful from a QA/QC perspective, which we think will be very desirable for clinical grade cell therapy products. These past couple of slides have really showed you how we're doing image-based machine learning and laser processing to produce high quality cells and tissues. And sometimes maybe there are some cells and tissues where we need some additional capabilities to make that cell and tissue. So we do have other capabilities that we use sometimes when using a small molecule protocol for differentiation, for example, is not enough. So we have in-house a computational framework that you can feed in single cell RNA sequencing data to analyze which genes should be turned on and off to drive a faster differentiation process. And for the most part, we work with small molecule differentiation approaches, but we've also experimented with CRISPR activator-based differentiation, which was really nice. So a dopaminergic neuron protocol, we were able to cut that timeline down to half of the time it would have taken with a small molecule approach. We've also done pattern delivery of cargo to cells, which is a very unique feature of the Cellino platform and can be used for multiplexed cell manipulation and gene modulation and those types of applications. The Cellino platform is very broad. There are many interesting features, but really what sets us apart is that we're the only technology that's capable of doing closed cassette manufacturing with single cell precision. And we really are focusing on that because it is a necessary step to making autologous cell therapies a safe and economically viable option for patients. We're, of course, borrowing very heavily from the semiconductor industry because large-scale optical manipulations have been developed there for decades now, and they've been able to scale things up tremendously. So we're borrowing those ideas and concepts and bringing them into how we manufacture iPSC-derived therapies. It's super exciting and compelling. And we hope that our unique approach will greatly enhance our industry's ability to serve patients. To that effect, let's talk a little bit about the business model. Our mission as a company is to deliver as many iPSC-derived cell therapies as we can to patients who need them. And to that effect, we've come up with a full stack business model where on the one hand, we related to clinical iPSC generation, we're partnering with biopharma and clinical stage academic groups to develop and deliver clinical grade iPSCs that can be used in their programs. And on the other end, we're also evaluating certain cell types in specific indications where the Cellino platform may have a unique advantage when it comes to using machine learning based approaches and recognizing which cells have the greatest potential for being the transplanted cells. Our team is split across many different areas. So we have optics, experts, biology, automation, and machine learning. We're thrilled to be working with Bob Pillay and George Church as our advisors. And I've been attending the MESA meeting for the past few years, and it's such a highlight every year, even though I know this year it's going to be virtual. I'm still really excited to connect with all of you. My co-founder, Dr. Marina Madrid, is also attending. So please do reach out to us, and we'd love to talk to you if you're in the iPSC cell therapy space. Our mission at Cellino is to automate the process of autologous iPSC cells and tissues and the manufacturing piece of that. And we're doing that in a very unique way by merging machine learning with laser physics and stem cell biology. Thanks so much for watching the talk, and I look forward to talking to you more at the conference.